Okay. All right. Yeah. Okay. We are streamed to YouTube right now. Okay. All right. I think we should wait a little bit more until people log in. So let me see. Okay. All right. So Bobby said, yeah, we are live. Yeah. Okay. All right. Did you overlap with Ja Hu in John Rubinstein's lab?
Ja who?
Yeah.
Yeah.
Oh, okay. I used to be a lab mate with him, so we did thesis lab in the same lab, Edmund
Oh,
McGee's
cool.
lab. So
UCI?
at UCI, yeah.
Oh, cool.
How is he doing? I haven't heard back from him a while.
Dan is here. Dan probably knows better about Ja's status.
He's
okay.
I don't know his title anymore.
I don't know. He's still in John's lab.
Oh, okay. Awesome.
I feel it's more like a specialist now.
I think he's graduated to a specialist now.
Yeah.
I see. Okay, good to know.
Yeah.
All right. So let's give some more minutes, and once I get more people logging, I'll get started. Okay. All right.
Sounds great. Dan, make sure you stop me when I start talking nonsense. I'm having fever right now.
You'll be fine.
Yeah, because I got a cold 3 days ago, and I thought it was like a random small cold, and then I started having fever this morning. I was just like, ugh.
Hopefully it'll go away from that.
Hopefully it's not COVID.
Hi, everyone. It's over past 10, but we are still expecting more people to log in. So let's give some more minutes. Hi, everybody. So let's get started. I think people are still logging in, but it's over 10, so I like to start. Thank you for your participation. We really had high quality talks from trainees, and I'm very excited to host another fantastic speaker today, Dr. Emily Ling-Lin Pai. She is currently transitioning to University of Michigan as a postdoc fellow. Today she will talk about her PhD projects in John Rubinstein's lab at UCSF. So just general logistics. To ask questions, please feel free to raise your hands using the Zoom feature, and I can point that to the speaker or type in the chat box, and I can present it to the speaker. Emily, please take it away.
Okay. Can you guys see the screen okay?
Not now. I did see it just a minute ago.
Oh, sharing is paused. Interesting. Let me do this one more time.
Sure. Okay, now I can see it.
Can you see this now?
Yes. Okay.
Yes.
So, thank you so much. Thank you all for offering me the opportunity to talk about my work today. So today I'm going to use the majority of the time to talk about my PhD work that was done in John Rubinstein's lab at UCSF. And then I will try to spend the last 5 minutes talking about what I've been doing at the University of Michigan here in Ann Arbor. So the title of my talk today is MAFB and CMAF specify cell and regional fate of paleo interneurons. So I believe probably the majority of the people here are interneuron experts, but for people who don't know about interneurons, in the mammalian brains, 80% of the neurons are excitatory neurons. So when they fire, they help us generate movements, thoughts, and then conduct memory formation. But if you imagine all of these neurons are firing at the same time, we're likely going to get seizure. And so this is when interneurons comes into play. Although they only have 20% of the volume, they actually form delicate synapses onto the excitatory neurons to make sure that they're firing in a time and location specific manner. Let's see. So interneuron development has become a very popular study topic over the past 10 to 20 years. Mainly it's because that the interneuron pathology has been reported to be implicated in many diseases, such as seizure, schizophrenia, and autism spectrum disorder. So some of the suspected pathophysiologies include that the patient either acquired de novo mutations or they acquire mutations from their parents. And these mutations, for some reason, will either lead to the interneurons to overfire, or they just fail to mature. And that means that they will not be able to generate the proper inhibitory signals. And so all of these will lead to the imbalance of excitation versus inhibition. And that's when we start to see symptoms in patients. So we think that it's very important if we can understand how interneurons are born and develop normally. That way we can, try to pinpoint specific checkpoints for therapeutic development. For my thesis projects, I mainly focus on a region, which is a transient brain structure called the medial ganglionic eminence, which is labeled in short as MG here. So this belly-like structure, it contains a lot of stem cells that generates around 70% of the total interneurons in the neocortex. And within this MG-derived interneuron population, we can roughly divide them into two parts. One is somatostatin expressing, and the other one is parabumi expressing. So somatostatin interneurons, they are born in general earlier, peak at around embryonic day 12.5, while parabumi interneurons, they are born in general later, peak at around E15.5. This is a very busy slide, but basically I'm just trying to tell you that, over the past 20 years of work, we now know that, the pan-MG fate is determined by a very complicated transcriptional cascade. But in terms of what molecule that's telling the MG stem cell to say that, hey, at this specific time point, you should start generating proper beaming interneurons rather than somatostatic interneurons, it still remains unclear. So one of the main mission of my project is to try to figure out if MAFB and CMAP have certain functions in mediating this PV versus SST fate determination process. So what are MAFB and CMAP? They are part of the large MAP transcription factor families, and they are the only two large MAPs that are expressed in the central nervous system. While we have limited amount of knowledge for the functions in the nervous system, they are actually very well studied in many other organs, including in bone, epithelium, macrophages, and pancreas development. So studies have suggested that MAFB and CMAP can either function alone or together to control cell fate determination and differentiation. So this is what we've known so far in the nervous system. So in the central nervous system, MAFB is known to control embryonic hindbrain regional patterning. So that being said, if an embryo for some reason is acquiring a loss of function MAFB mutation, when these babies are born, they're going to be born lethal because they cannot breathe when they're born. In a more recent report, it's suggesting that MAFB is playing important roles in a very small subset of somatostatic interneurons for their axonal targeting. In the peripheral nervous system, we know that MAFB promotes the auditory ribbon synapse formation, while CMAP is regulating the touch receptor differentiation. So why do we care about MAFB and CMAP in interneurons? The reason is because that we have known that the expression of MAFB and CMAP are enriched in the MGE for a long time. So in the top panels, you're looking at the RNA in situ hybridization probing the MAFB and CMAP RNA expression at E15.5 wild-type tissues. You can see that both MAFB and CMAP expressions are enriched in the MGE. It also labels the migrating interneurons that's passing through the CGE and also among the ones that are actively migrating in the immature neocortex. When we use the antibody to probe the MAFB and CMAP protein expression at the same age, you can see that the antibody beautifully labels the immature migrating interneurons in the brain. So with this consistent and persistent expression of MAFB and CMAP, we know that they must have some sort of functions during the interneuron development. So to study their function, we took a genetic approach. Here we are using the NKX 2.1 Cre, which is a Cre line that's very specific to the MG lineage. So we crossed the NKX 2.1 Cre with the MAFB and CMAP flux allele to generate the MAFB and CMAP single and double mutant animals. And within the crossing, we also introduced AI14, which is a Cre-dependent reporter allele. That means that, whether or not we're generating wild-type animals or mutant cells, all of these cells will be labeled with the red fluorophore. This will facilitate fate mapping and tracing of these interneurons. But then the basic idea is that we just wanted to know when we delete MAFB and CMAP in the MGE, what is going to happen downstream. So when we generated the mutants, first thing we wanted to know was, is there any phenotype? So we decided to look at the adulthood first. So we harvested the wild type of mutant somatosensory cortex and quantified the red positive cells that are supposed to be MG-derived interneurons. Compared to the wild type, you can see that in the double knockout neocortex, there's a drastic reduction of MG-derived interneurons, which is supported by the quantification here, showing that the double knockout is having 70% loss of MG-derived interneurons. Single mutants, they seem to only have intermediate phenotype. So this figure alone tells us two things. One is that both MAFB and CMAP are required to generate the proper amount of interneurons in the neocortex. And then during this process, MAFB and CMAP likely are playing compensatory roles because only when we delete both of them, that's when we get the biggest phenotype. So we move on to look at the MG-derived interneuron subtype markers, parabimine and somatostatin. We observed that in the double knockout, within the remaining surviving red cells, there's a less proportion of them that are expressing HARBP, suggesting that in addition to the huge cell reduction in the double knockout, these mutants also have preferential loss of PV interneurons. And we did not see that type of phenotype in the somatostatin population. So when we first collected these data sets, our first thought was, where did the cells go? So we try to survey different possibilities that will lead to the cell reduction phenotype. So first is that we use the IBA1 to probe microglia prenatally in the MGE and then postnatally in the immature and mature cortices. Surprisingly, we did not find any differences between wild type and mutants, suggesting that the microglia engulfment is not the major cause that's leading to the cell reduction. So we wonder that if losing MAFB and CMAB somehow would lead to the increased suicidal activities in these interneurons. So we used tunnel assay and cleave caspase 3 as two independent assays to look at the apoptotic activities across developmental ages. And again, we did not see major changes between wild type and mutant. So up to this point, we know that it's not microglia engulfment or increased apoptosis that's leading to the cell reduction phenotype, but what's really happening perinatally and postnatally that's leading to the cell reduction, we still don't know. But we thought maybe we can go back to embryonic stage and ask, when the MGs themselves are lacking MAFB and CMAP expression, are they also losing their capability of generating interneurons? To do so, I utilized a nucleotide analog, which is called EDU. When we inject this EDU into the animals, it can actively label the actively dividing stem cells. So I injected the EDU into E12.5 embryos, incubated for 6 hours, which is considered 1 cell cycle length for the MGE stem cells. I harvested the tissues and co-stained with the EDU and TUJ1. So the purpose is that I wanted to know within 1 cell cycle length, how many of the EDU labeled MG stem cells will move on to become post-mitotic neurons that would be labeled by TUJ1. And I used this as a neurogenesis index. And very surprisingly, I found out that the double knockout had a 2-fold increase of neurogenesis activity, which is pretty counterintuitive considering what we saw in vivo. So is it really the case? We then probed the somatostatin RNA expression at both ages at E15.5 at a P0. The left panels are both coming from the wild type tissues, and the right panels are both coming from the double knockout tissues. From here, I hope it's very clear to you guys that at both ages, double knockouts seem to have way more somatostatin interneurons in the neocortex and also in the immature hippocampal region. So earlier I mentioned that the somatostatin interneurons, they're usually born early, peak at around E12.5. So to directly quantitate if the early born stem cells are really generating more somatostatin interneurons, this time I modified the EDU pulse chase experiment. Again, I injected the EDU at E12.5, but this time I let it incubate for 3 days. This would allow the stem cells to quit cell cycle, move on to do neurogenesis, and allow time for the interneurons to migrate to the neocortex. So 3 days later, I harvested the animals and I co-stained with somatostatin and EDU. Sorry, I'm having a cold right now. So very consistent with in situ finding. Again, in the neocortex, in the double knockout, we found out that more of the somatostatin interneurons were labeled with the EDU. So this was the direct evidence to suggest that the double knockout MGs themselves are generating more somatostatin interneurons. So while not knowing the molecular mechanism, at least we now know that MAFB and CMAP likely are functioning together in the MGE to repress this somatostatin interneuron neurogenesis. And I will be addressing a little more about the molecular mechanism later on in the presentation. So to try to study these mutant cells in a more systematic way, meaning that we don't just care about their marker expression, we don't just care about their cell numbers, we also care about if they fire properly in the adulthood. To do so, we collaborated with the Jean Paul's laboratory at UCSF, where they were able to harvest the adult animals, generated acute brain slices, and then patch onto these red cells in cortical layer 5. And this is what they found. Surprisingly, that the double knockout cell seems to fire pretty comparably to the wild-type counterpart. But MAFB single mutant, they seem to have reduced EPSC amplitude. This would suggest that, when the interneurons are lacking MAFB expression, it probably will lead to some sort of postsynaptic anti-receptor component changes that's leading to the amplitude changes. CMAF single mutant instead, they have increased EPSC frequencies. And this would suggest that, when interneurons are lacking CMAF expression, for some reason, these cells will start to receive more presynaptic stimulation that's causing them to fire more frequently. So to support these physiology findings, I did 2 follow-up experiments. One is that I harvested these red cells from the P0 animals, cultured them in dish for 14 days. And then I used PSD95 and VGLUT1 to localize the excitatory synapses that are formed onto the proximal dendrites of these interneurons. And this is what I found. In the MAFB single mutant, there was a trend of decreased amount of excitatory synapse. But in the CMAF single mutant, they seem to have way more excitatory synapses that are formed onto the proximal dendrites. And we think that this likely is one contributing factors that's leading to the increased EPSC frequency phenotype. I also surveyed the morphologies of these interneurons. So MAFB single mutant, their morphology didn't seem to differ that much compared to the wild type. But CMAF single mutant, they have way more neurites growing out from the cell body. And we think that this could be another reason why they're firing more frequently, because they tend to have more neurites, meaning that they're going to have higher cell surface area, which will increase the probability of them receiving presynaptic inputs. On the other hand, double knockout cells seem to have neural algorithm defect for a reason that we don't know just yet. So for this part of the story, we know that during the interneuron maturation and morphology, and also in building their electrophysiological properties, MAFB and CMAF are likely playing different or opposite roles to control these processes. So I've described many phenotypes in these mutants to you up to this point, but what we care about is the mechanism. What is driving these phenotypes? To try to figure out the mechanism, we utilized single-cell RNA sequencing. And the mutant that we're targeting here was mainly focusing on the double knockout because this is the mutant that gave us the most phenotypes. I targeted at an age at P0. The reason is because one, P0 is the age where the cells are very viable. So is the golden period where we can get the best quality cells for single cell experiment. Secondly is that P0 is the first time point where we started to see significant cell reduction phenotype in the double knockout. So to briefly describe how I did the experiment, I harvested the age and gender matched wild type and double knockout P0 animals, dissected out the cortical tissues, which would include the hippocampi. I gently dissociated the cells, loaded onto the 10x machine, followed by library preparation, sequencing, and data analysis. So I collaborated with the Weissman lab at UCSF and mainly used the Seurat pipeline to conduct the unbiased data analysis. So here you're looking at the 2D projections of this complicated data sets. Here I'm showing you 2 UMaps, both contain around 25,000 cells coming from wild type and double knockout animals. So to the left, the cells are color-coded based upon the cluster identity. So for here, you just have to trust me that the cluster 1 is coming from the MG-derived interneurons. And this is based on the marker co-expression, which include LHX6, MAFB and CMAP, neuroxophiline 1, and among others. To the right, the cells are color-coded based upon the genotype. And I hope this is very clear to you that there's only one cluster that has a clear separation between wild type and double knockout, which happens to be the MG-derived interneuron population. So we decided to take these cells and move on to conduct differential expression analysis. So serving as very good internal control, you can see that in the double knockouts, there's a huge decrease of CMAP and MAFB expression, which makes sense. There are also some other genes that are differentially expressed. For example, MEF2C was downregulated, and consistent with the RNA in situ finding, the somatostatin expression was increased. There's also increase of PNOC. We also surveyed some, general interneuron markers, including DLXs, GAT genes, and among others, and we did not see a major change between wild type and double knockout. So this suggests that, the double knockout, when the cells are lacking MAFB and CMAP, they for sure are having some property changes, but they still remain that, the general interneuron fate. So the most important thing after a sequencing experiment is for validation. For the time's sake, I'm just going to be talking about 2 genes today. The first gene is MEF2C. This is a gene that was identified to be downregulated in the double knockout. And here we choose to use the MEF2C antibody to probe the MEF2C protein expression for validation. And the main reason is because MEF2C is very widely expressed, both in excitatory and also inhibitory neurons. So we decided to just look at the MEF2C expression in the red positive MG-derived interneuron population. So here you can see that in the P2 wild-type neocortex, the majority of the red cells are having MEF2C expression. So which would be the green dots in the middle of the red cells. In the double knockout, you should see 2 things. One is that the double knockout has a drastic cell reduction phenotype in the red positive cells. And among the remaining surviving red cells, a lot of them don't have MEF2C expression, which is consistent with the seq finding. There are several reasons we care so much about MEF2C. One is that MEF2C was shown to regulate synapse formation and morphology maturation for excitatory neurons, but their function for interneurons remains unknown. And 2 years ago, there was a report to suggest that MEF2C was actually very important for PV interneuron development. So they used the DLX5-6 CRE to delete MEF2C. And in their conditional mutant animals, they found out that these mutants had a drastic reduction of PV interneurons. This should sound familiar because this is exactly what we observed in the double knockout animals. So we wonder if it's possible that MEF2C is genetically downstream of MAFB and CMAP that's driving the PV interneuron production. So to test this idea, we utilized the modified MG transplantation assay. So the animal here I'm using is the MAFB, CMAP, and AI14 triple flux animals. So I harvested the MG stem cells at E13.5 from these animals. I dissociated them in the tube. Within one tube, I will introduce the CRE expressing vector. That way I can generate the double knockout cells in the tube. In another tube, I would co-transfect the cells with CRE expressing vector and MEF2C expressing vector. That way I can artificially generate double knockout cells in the tube, now having MEF2C expression. So after modifications, I transplanted these cells back into the P0 wild type animals and let them grow for 30 days before I evaluate their interneuron phenotypes. So when we look at the wild type and double knockout transplanted cells, you can see that, again, in the double knockout, they had a preferential loss of PV interneurons. This is consistent with what was observed in vivo. And you can also see in the representative image that these double knockout cells, they're lacking the PV expression. When these cells were reintroduced with the MEF2C expression, it partially rescues the PV expression, again shown here in the representative image. So this data set suggests that MAFB and CMAF likely are promoting MEF2C expression in order to drive PV interneuron production. Earlier, I mentioned that the double knockout cells, they also seem to have neurite algorithm defect. Since MEF2C is able to regulate morphology in excitatory neurons, we then wonder if it's possible that MEF2C can rescue this double knockout neurite algorithm defect phenotype. So I did a similar in vitro culture experiment where I harvested the wild type and double knockout red cells from the neonatal neocortex. And I grew them in vitro for 14 days. But with one of the double knockout wells, one day after plating, I would transfect them with the MEF2C expressing vector. That way I can evaluate the MEF2C rescuing effects. So 14 days later, I look at their morphology. Again, the double knockout cells, they seem to have neural algorithm defects consistent with our previous finding. But when these cells are reintroduced with the MEF2C expression, their neurites now are growing in a healthier pattern, which is supported by the show analysis quantification result. But morphology maturation, this is a thing that oftentimes happens postnatally, but the previous experiment was done using NKX 2.1 Cre mutant, meaning that the MAFB and CMAF are deleted very early on. Here with the morphology maturation, we care more about if MAFB, CMAF, and MEF2C would have postnatal roles in mediating the morphology maturation process. So to tackle this question, I actually modified the in vitro culture experiment. So this time I went back to utilize this MAFB, CMAF, and AF14 triple flux animals. Again, I harvested the cells from P0 neocortex, grew them in vitro for 14 days. One day after plating in one well, I would introduce Cre-expressing vector so that I can artificially generate the postnatal double knockout cells in vitro. And in another well, I would co-transfect Cre-expressing and MEF2C expressing vectors. That way I can generate the postnatal double knockout cells that now expresses MEF2C. And then 14 days later, I will evaluate their morphology. A little different from the NKX 2.1 Cre result is that these double knockout, postnatal double knockout cells, they didn't seem as healthy as the NKX 2.1 Cre double knockout mutant. Their cell bodies tends to be smaller, and again, they also had neural algorithm defects. But when these cells are reintroduced with the MEF2C expressing vector, their neurites were able to grow in a healthier pattern and sometimes grow even more than the wild-type counterparts. Although the result is a little different from the NKX 2.1 Cre result, but both experiments suggested that the MEF2C was showing the rescue effects. And so when I saw this rescue effects, one thing that bothered me a lot was that, is this rescuing effects a direct rescuing thing, or is it something that's not cell autonomous? So luckily, right before we submitted the paper, a former lab member was able to perform the MAFB ChIP-seq data, where she was able to show that MAFB had a strong binding peak in the MEF2C promoter region. So this is really the direct evidence to suggest that at least one of the MAFs is directly regulating the expression of MEF2C. So to summarize this part of the story, I hope the data can convince you that prenatally MAFB and CMAF are promoting the expression of MEF2C in order to drive PV interneuron production. And postnatally, we think that MAFB and CMAF may be still maintaining the expression of MEF2C in order to mediate the morphological maturation process. The second gene that I'm going to talk about is the pre-pronoseptin. This was a gene that's found upregulated in the double knockout. It also had a strong promoter binding peak by MAFB. This is a gene that was actually pretty understudied. 10 years ago, a paper from Stanford suggested that this could be a interneuron marker. So they simply did the PNOC labeling with GAD and found that they're colocalizing, but that's about it. Like no more story after that. So before we move on to look at what's happening in the double knockout, we first really have to validate, the wild type expression of PNOC. So we did traditional RNA in situ on PNOC at P0 wild type tissues. If you look at the paleo regions, which include the neocortex and hippocampus, you can see that the purple punctate, which represents the PNOC RNAs, are more enriched in the hippocampal region. When we use the dual color fish with the red labeling the MG-derived interneurons and then the green labeling the PNOC RNAs, you can see that a lot of the MG-derived hippocampal interneurons are expressing PNOC. But the PNOC expression in the neocortex seems to be minimal to none. So these two data sets suggest that PNOC can be a good marker for hippocampal interneurons. We move on to look at PNOC expression in the double knockouts. Here, it clearly shows that the double knockout hippocampus has way higher expression of PNOC. There was also ectopic expression of PNOC in the dorsal medial part of the neocortex. And again, using two-color fish, you can see that in addition to the increased expression of PNOC in the hippocampal MG-derived interneurons, there's again ectopic expression of PNOC. So these data sets leads to our hypothesis where we think maybe MAP double knockout is generating more hippocampal interneurons that are expressing PNOC. So to test this hypothesis, this time we modified the post-chase experiment. So this time I used two different nucleotide analogs. One is called BRDU. I injected at E12.5 to label the early born stem cells. And then at E15.5, I will inject EDU to label the later born stem cells and later born interneuron population. I allow them enough time to become interneurons and migrate to the neocortex and hippocampus before I evaluate their co-expression with allergic 6. So let's first look at the early born population from the MGE, which should be labeled by allergic 6 and the BRDU. From here, you can see that in the double knockout, there are clearly way more allergic 6 positive MG-derived interneurons that's populating in the double knockout hippocampus. And a lot of them are BRDU positive. So this is suggesting that the double knockout MG stem cells are generating more interneurons at E12.5 that will later on become hippocampal interneurons. And out of curiosity, we actually went back to look at the E12.5 generated cortical interneurons and found out that at this age, double knockout is actually generating less cortical interneurons in a way that this data set is implying that maybe the double knockout MG stem cells are generating more hippocampal interneurons at the expense of cortical interneurons. And we found similar results in the later born population where they should be labeled with allergic 6 and EDU. So again, when you look at the P2 double knockout hippocampus, it's well populated by the LX6 positive interneurons coming from the MGE, and a lot more of them are EDU positive. So both data sets together suggest that the double knockout MGE is generating more interneurons that are going to become hippocampal interneurons, regardless of the developmental time points. So in other words, we think that MAPB and CMAP likely are repressing the production of hippocampal interneurons in the MGE. So to quickly summarize what I've shown you today, I hope I can convince you that we think that MAPB and CMAP are important in balancing the production of PV versus SST interneurons. They do so by prenatally promoting the expression of MEF2C in order to drive PV interneuron production. And I've also shown you data to suggest that MAPB and CMAP likely are maintaining the expression of MEF2C, that has functions in mediating their morphology maturation. More excitingly, in addition to this PV versus SST fate determination, this likely is the first report to suggest that there are actually transcription factors that are existing to control the balance of amount of hippocampal versus cortical interneurons that should be generated by the MGE. And through the seq data, we also found that PNOC can be a good marker for hippocampal interneurons. And so usually when I presented my data sets, a lot of people would ask me, your double knockout seemed weird. They had a lot of cell reduction. They have preferential loss of PV interneurons. They also have some other phenotypes, but do they have seizures? So we continued to collaborate with the POS lab at UCSF, where they were able to record the spontaneous EEG activities from the NKX 2.1 Cree generated wild type of mutant animals, and found out that the double knockouts, they indeed have seizures. So when they broke down the EEG firing patterns, they seemed to think that these epileptic activities in the double knockout are more similar to the absence seizure-like activities. So this is the time when I, left the lab and the project, but they are still actively trying to pursue this project and then try to use physiology tools to figure out what could be the main driver of the epilepsy phenotype in these mutants. So while they continue to use the physiology tools, in the last 5 minutes, I'm gonna propose a new methodology, which is something that I've been doing at UMich that can be utilized to visualize the neuronal circuitry. So in my current lab, we do something called the spectral connectomics. This is a fancy term, but in lay people term, basically we're trying to say that, we are modifying the brain tissues in a way that we can, do many rounds of immunostaining, and we can use this fluorescent spectral information to collect information not only in anatomy, but also in neuronal morphology, their molecular markers, as well as their synaptic connectivities. So we do so by incorporating a technology called the tissue expansion. When I was in the lab, I was able to modify the original MIT protocol. So now we utilize a special proprietary gel recipe where we can now clear the brain sections and also expand the brain sections at the same time. And so this allows different things. One is that as the tissue gets expanded, while we're still using the same confocal microscope, because the physical expansion of the tissue actually increased the resolution of light microscopy. And another thing is that because now that the tissues are well anchored in the hydrogels, we can treat this like a SDS page object, which in theory allows us unlimited amount of immunolabeling. So for example, if we're gonna look at how interneurons are connected with each other, we would need interneuron morphology information, their subtype markers, and their synaptic markers. So here we can easily throw in first round of staining with interneuron markers. Here you're looking at 3 examples, including PV, seminostatin, and calbindin. We can easily strip out these immunostaining markers and then throw in another round with synaptic markers. So you're looking at Jeffrin, Homer, and Bassoon staining, and you can see that the resolution is really high. We can easily identify inhibitory versus excitatory neuron synapses onto these tissues. We combine tissue expansion with the Brainbow technology, and the reason is because we wanted to know each individual neuron's morphology in a detailed manner. And if we just use regular GFP to do staining, oftentimes the neighboring cells, they would all have GFP label processes, which would complicate the morphology reconstruction processes. So Brainbow is a special fluorescence protein expressing vector in a way that, when we, let's say, inject into a Cree-carrying animals, it allows dense labeling of the neuronal populations with each individual neighboring cells being labeled with different colors. And this makes the morphology reconstruction way easier. So if we're gonna solve the interneuron connectome, we need morphology information. So if you imagine, I perfectly overlay this rainbow label morphology information together with a molecular marker and synaptic marker. This is the result that we were able to publish in Nature Communication. We showcase that, in the somatosensory cortex across different cortical layers, we know how interneurons are cross-talking with each other. We know each individual interneuron's morphology, their molecular markers, subtype markers, and then we know exactly where excitatory and inhibitory synapses are formed onto the cell bodies. And so my current project is to try to scale this up, hoping to achieve this in a whole brain scale. And in parallel, I'm also using this to apply to epilepsy and autism animal models. And we think that this is a very good way to visualize how synaptic connectivity and neuronal morphology can be disrupted in the disease context. And soon in the future, I hope that you can also be able to appreciate that we're gonna generate a autism animal model connectome atlas, hoping to be a good complement to the physiology findings in the research community. So with that, I would like to thank my former lab for the mentorship and support for the math project, the POS lab for the physiology work, and then my current lab at UMich, which is funded by multiple Brain Initiative grants, which allow me to explore fancy technologies. Thank you again for your time, and I'm happy to take questions.
Thank you, Emily, for fantastic talk. Is there any question from the audience? You can unmute yourself or type into the chat box. Maybe I can start with my question then. Yeah, that was really thorough characterization, and I was very impressed by very thorough molecular characterization of those double knockout mutants. So I have one clarification question. So it looks like the double knockouts, they have increased SST and hippocampal interneurons, but overall in the cortex, the 70% of the interneurons are reduced. Is that what you calculate? So my question is, when you quantify, do you think increase in SST and hippocampal interneurons, that's kind of add up to like overall 100%? Or is there any other phenotype because SST and PV obviously just two kind of oversimplified interneuron classes, and do you think there's any other defects in other type of interneurons?
So if you're looking at the dilution of MAP-B and CMAP and other types of interneurons, which are supposed to be coming from, like, let's say POA or CGE, they are not affected. And then, yes, indeed, we see increased somatostatin interneuron production early on in the hippocampus. And it's really surprising to us that when we look at this in adulthood, they also had a drastic reduction. And so that increase of somatostatin interneuron population was transient, and we still don't know like what's leading to the cell reduction postnatally, both in the neocortex and in the hippocampus.
And you didn't see any cell death defects there, so it's still unknown.
It's still unknown.
Okay. All right. So I see a question from Jasmine. Would you like to ask Emily directly, Jasmine? I can present it to you. So question from Jasmine. Thank you for your research. Very informative, and the level of explanation was great. Amazing presentation. I hope your research continues to expand in the direction you are hopeful for. Okay, I think it's a kind of a comment from Jasmine. Thank you.
I have a question.
Okay.
Really nice talk, Emily. Good job. I was kind of curious, a technical question about the when you did the rainbow tracing. Was that like a manual segmentation, or did you automate that?
We did semi-automatic tracing. So our lab developed a N-tracer, which is a Fiji plugin. So it's a little similar to the Neural Lucida, but then it still requires that you to manually go through the Z-stack and then make sure that each neuronal processes are going in the direction that you want, and then record them in order to do reconstruction. But it can be semi-automatic.
Okay. Is that online? That plugin?
It is, and then we have several YouTube tutorial videos for new users to refer to.
Great. I'll check that out. Thank you.
Any other questions?
Yeah, Dr. Reback, it was very exciting talk, and it's terrific work from the Rubenstein lab. I was very much surprised that these animals developed seizures, and I was wondering when Dr. Paz and collaborators established an age when the seizures began. Do you know?
This is a very complicated question. So we sent over the animals when they hit like a young adulthood age, but with the implant settings and such, they weren't able to record them until the animals were like close to 1 year old age. And so that was the only age where they saw the seizure activities. And we don't have any data to suggest that these seizure activities happen early on. And I don't know if they have like new data to support early onset seizure activities. But what I can tell you is that Dan and I, we handled these mice for years. We never saw like tonic-clonic physical seizures. And so I would believe that when they said this is more like absent seizures, that could be true.
So they were definitely electrographic because you showed the different sharp wave, I guess, right? Okay. Well, thank you, and wish you good luck and continued luck in your studies at Michigan.
Thank you so much.
Is there any other question from the audience?
I have a question.
Okay. Yeah,
Hi,
please.
can you hear me? Very nice
Yes.
to talk, Emily.
Thank you so much.
I still don't really understand the mechanism by which this transcriptional regulation occurs only for parvalbumin, differentiation. So is MEF2C not expressed in somatostatin population?
That is a great question. I didn't look at it, but I think the main what is the word? I'm losing the word. So the two major claims coming from our lab and Gore Fischel's lab is both coming from the deletion, the mutant animals. And so, when they delete MEF2C, they saw a huge reduction of PV interneurons. And that's how they started to claim that MEF2C is important for PV interneuron development. But none of us have actually looked into, whether or not MEF2C is expressed in the somatostatin population. And that's something I should have done, and I didn't. I'm sorry.
Okay, thank you.
I don't have any questions, but I just want to say great job.
Thank you
As
so much,
always.
Dan.
So I have one question related to the previous question. So it was wonderful that you could actually rescue PV interneurons by expressing MEF2C and you transplant back into the brain. Is there any critical period for that to kind of rescue the cell phenotype?
That I'm not sure because I only tried one age. But I think one thing we could do is that, we can do sequential transplantation assay from maybe like from E11.5, 12.5, 13 to 15 to see if the rescue effects was really only existing at that, E13.5 age.
Okay. All right, thank you. Any other question from the audience? Okay, I think with that, let's virtually thank Emily for a wonderful talk. And okay, have a good day. Bye.
Thank you so much.